Hello everyone, it's Andrea Guerra and I'm a postdoc clinical research fellow in Sapienza University of Rome, and I'm going to show you the results of our study recently published on brain. Although bradykinesia is the cardinal motor symptom of Parkinson's disease, its pathophysiological mechanisms are still unclear. It is well known that there is altered oscillatory activity within the basal ganglia in PD, and in particular, there is an increased beta oscillations and reduced gamma oscillation, which are putative mechanisms involved in bradykinesia pathophysiology. However, recent evidence suggests that bradykinesia is a network dysfunction, and the primary motor cortex node of the network plays a relevant role in generating this symptom. And thus, we investigated whether the oscillations at the primary motor cortex level play a relevant role in bradykinesia in patients with PD. We decided to approach this issue by externally modulating the M1 oscillations through non-invasive brain stimulation. And in particular, we use transcranial alternating current stimulation, which uses an oscillatory patterned electrical current, which is able to entrain brain rhythms and enhance the power of the targeted frequencies. We applied beta, SHAM, and gamma TACS in a randomized manner over the primary motor cortex during a repetitive finger tapping task, which was monitored by kinematic techniques, and we measured several kinematic parameters. Also, in the second part of the experiment, we used transcranial magnetic stimulation techniques to verify whether corticospinal excitability or the activity of inhibitory interneurons within M1 changed during beta, gamma, or SHAM TACS. All patients underwent 2 sessions, one in the off and the other in the on condition. Our results show that beta TACS decreased movement velocity, and so beta TACS worsened bradykinesia. In contrast, gamma TACS increased movement amplitude, so it produced a beneficial effect on bradykinesia. Importantly, amplitude and velocity were symmetrically increased during gamma TACS and decreased during beta TACS. Also, beta and gamma TACS modulated psyche, and in particular, both stimulations reduce this intracortical inhibition of M1. Importantly, the effect of beta TACS on psyche was negatively correlated with the effect of the stimulation on movement kinematics. And indeed, patients with the greatest modulation of psyche during stimulation were those in which beta TACS had the less detrimental effects on movement. Also, we found a positive correlation between the effects of gamma TACS on psyche and on movement kinematics. And in this case, patients with the greatest modulator effect on psyche were those in which the stimulation produced the greatest beneficial effect on movement. Finally, we found that the modulatory effects of TACS on movement kinematics and TMS measures was comparable between off and on medication conditions. And to conclude, our results suggest that beta and gamma oscillations at the primary motor cortex level play a relevant role in bradykinesia pathophysiology. In particular, increasing beta oscillation in M1 favors bradykinesia, while enhancing gamma oscillations counteracts bradykinesia. And thus, the role of beta and gamma oscillations in M1 is similar to that of these oscillations observed at the basal ganglia level in previous studies. And we thus may hypothesize that entraining beta and gamma resonant neurons in M1 enhance these rhythms in the entire basal ganglia thalamocortical loop. Finally, our data support the hypothesis that cortical disinhibition is a possible compensatory mechanism against bradykinesia and PD. Indeed, we found that cortical disinhibition counteracted the pro-bradykinetic effect of beta and promoted the anti-bradykinetic effect of gamma. Thank you for watching.